suis ravi d'accueillir Matthieu JENSEN, qui est un grand spécialiste de rotavirus. Alors, c'est ma manière, parler par la place de ma manière d'inviter les gens, de vous faire un séminaire. Ça n'est pas quelqu'un que je connais. Et ça n'est pas une relation. Moi, je choisis les gens qui sont les plus cités, les plus dynamiques par leur production scientifique. Et je les invite, et la plupart du temps, comme je l'ai, ils ont la gentillesse de me dire oui, et de venir voir ce que nous on fait aussi, ce qui nous permet de discuter avec eux. Et la plupart des très bons orateurs, des meilleurs orateurs que nous avons eus ici, sont des orateurs qui ont été choisis sur leur production scientifique alors qu'on ne les connaissait pas. Et donc, la plupart des séminaires que vous voyez des séminaires qui sont par les stars des maladies infectieuses mondiales. Et donc, c'est comme ça. Et on verra, j'espère qu'on ne sera pas déçus, that we will not be disappointed, but usually by selecting the highly cited people and the people very, very productive in their own field, we are always happy with it. So he's not so far, he's not a friend, he's not a relation, this is the first time that we meet, but we identify him by the quality of his science, and I hope that it will transfer his way of thinking and doing science to you that need to learn how to be as good as he is yet. Okay? Yes.
Right. Thank you very much for this very nice introduction. So, as Didier mentioned, my main background was work on rotavirus. So actually, I'm a bioengineer, so I don't have a medical background. And actually, so rotavirus and viruses causing gastroenteritis. And actually, from that we morphed into actually viral metagenomics, initially of stool samples, initially of fecal material, and then also in other types of samples. So what I would like to tell you today is about the role of the virome in human health and disease. And as you all know, in the last years, there's a lot of research being dedicated, focused to the microbiota. And there's also quite a bit of nice work done in the institute here. But very often these are mainly focused on bacteria. And of course bacteria are an important part, they're not the whole story. And what I would like to achieve today is kind of give you an impression about the role of viruses can play in human health and disease. And in some ways it might be quite obvious, but there might be other subtle, different ways you might not directly think about. So that's kind of the goal, to give you a bit of a flavor about what different ways viruses could influence our health. So as I said, microbiota is a hot research field, and probably one of the most dense locations of microbiota is actually the human gut, or where there's a very dense network ecology of different types of organisms. And so of course we have bacteria, but of course we have other players there, with archaea, protists, fungi, parasites, and virus. And pretty much anywhere you would look on your body or in any environment, you will find ecosystems of these different organisms living there together. So also if you would look at the human body, whatever part you would be looking at is like a different ecosystem of microorganisms. And this is already an older slide, but just gives you an impression. If you take different swabs from different places on your body, and you would look at, in this case, only bacteria, but if you look at these bacteria, see what is present there, you would see that even on the skin of one person, there's a great variety of different microorganisms living on these different places of the human body, as you can see by the different colors representing different groups of bacteria. You probably have seen these numbers before, so it's estimated that if you look at the human body, that the number of human cells you have is compared to the ratio of bacterial cells living in and on us is about 1:1 to 10:1, depending what citation you look at, with 10:1 being in favor of bacterial cells. And in addition to that, if you look at the number of genes these microorganisms encode, that's about a factor 100 times more than the genes our own human genome encodes. So there's a lot of potential there for these additional genes to be also used to our own benefit. And in addition, everybody's microbiome is unique. If I would look at your microbiome or the microbiome of your neighbor, there will be a lot of differences there. There's some things in common, of course, there's also a lot of diversity present there. So actually around the turn of the millennium, around 2000, 2001, the human genome projects were finished and people thought, oh well, this is amazing, now we have all these human genes and we're going to be able to explain a lot of the human diseases, which came true to a certain extent. There is still a lot of unexplained things that people could not explain based on the human genome. And now the next big thing is metagenetics, not only looking at the genomes of the humans involved or the organism involved, but also looking at the genomes of all the microorganisms that live in and on us and see how that could explain maybe some diseases. And as I said, now there is at this point a lot of research going on all over the world with huge projects like MetaHit or the Human Microbiome Project, looking in massive scale to these microbes. And there are also a lot of other studies going on where people have investigated the link between microbiota and a number of different human diseases. As listed here, there's probably not a complete list, but it gives you an idea about a large number of diseases where people are looking what the role of microbiota could be there. So okay, back to the microbiome. Of course, I already mentioned there are many more members than just bacteria. So how do we investigate the microbiome? Of course, we all know, I assume most know that bacteria and archaea, we use a 16S, which is a conserved region in the bacterial genomes, which are very conserved and can be used to identify bacteria. A similar approach can be used for protists, fungi, parasites, for eukaryotes, where we can target 18S, and as you might know, 16 and 18S also have a common origin already in cellular life, and this can be used to generate the tree of life. Very beautiful bacteria, archaea, eukarya, but also what you may or may not notice, there are no viruses on here. The viruses cannot be put in the tree of life simply because they don't have a homolog 16 or 18S, which can be used to compare them. Not even 16 or 18S, but actually there is no conserved universal region in viruses in general. And the reason is because viruses are not monophyletic. They didn't arise at one point, they probably arose on multiple different occasions during the course of evolution. So there's no conserved region we can use to look at the entire virome. That's also one of the main reasons that virome research has kind of been neglected with, compared to bacterial or also eukaryotic metagenomics. So before I will tell you a bit more about how we can analyze the viruses, can analyze the virome, why do we actually care about these viruses? I just want to give you some numbers. You may also have seen some of these numbers, but it's estimated the number of viral particles present in the world is about this number, 10 to the 31st, 10 to the 32nd, and that's mainly based on extrapolations from data from oceans, where they also calculate the number of viruses there and then extrapolate that. So that's an amount that's very hard to conceptualize, to think what that means. Somebody did the effort of calculating if we put all of these viruses on top of each other, how far would we go? And they came by a number which reached far in the nearest 60 galaxies in the universe, which corresponds to more than 4 million light years. Again, very hard to imagine, but again, a number to show you that these viruses are very important, not only for our health, but also for global ecosystems, etc. So they're very abundant, they play a very important role in evolution of life, but again, they're most of them are very poorly studied. So if we come back to humans, for example, if you look at the fecal sample, if you have 1 gram of fecal sample, there's an estimated 10 to the 8, 10 to the 9 viral particles that will be present there. In addition, these viruses are morphologically highly divergent, and they've become even more highly divergent due to the research done at this institute with the giant viruses, of course. This is just a selection of viruses, with of course here the Mimi viruses. So what I want to show you is that they're morphologically very divergent. So if viruses are huge, the Mimi viruses, 700 nanometers, now I think there are other viruses coming up close to 1,000 nanometers. We have, on the other hand, very small viruses, such as circoviruses, around 20 nanometers, and a whole area in between. We have spherical icosahedral-like viruses. We have viruses which are rod-shaped. We have bacteriophages in different beautiful forms. These viruses can have an envelope, they can have it can be without an envelope. So there's a lot of diversity in morphology. So that's another difficulty which is added to if you want to investigate the virome. In addition, they're one of the most abundant and fastly mutated genetic elements on Earth, because very often they lack any proofreading activity, so a lot of genetic diversity is generated when these viruses replicate in their respective hosts. Some viruses also, most of you will know that, can be considered as commensals. They're chronically infectious. There are probably many more than you would think of right now, and this is an older slide from 2009 from the lab of Skip Virgin, where they did, based on literature searches, they did some estimation of how many people were chronically infected with a number of viruses. So what you can see here is a number of viruses, and on the y-axis here is the estimated number of people infected with those viruses. So if you see here, this is in millions, so 10 to the 4th is 10,000 million. We're not there yet in human population, so but we're approaching that, and as you can see here, some of these viruses are believed to be present in everybody. Everybody has these viruses in them without being sick. So if we want to quickly go over some of them, of course, endogenous retroviruses, so some people consider that part of the viromes, other not. It's a matter of, you can debate that. So these viruses we all have. They're inherited from our parents. They're part of our genome, and actually a large part of the genome we have is derived from endogenous retroviruses, which inserted their genome in the host genome in the past. And then another group which is quite interesting are anelloviruses. Anelloviruses only have been discovered also around the year 2000. And these are very small viruses. They have single-stranded circular DNA genomes. They're about a few kb small. So they're very small viruses. And they actually were found initially in human blood. And now when people have looked, pretty much everybody will have in his blood these anelloviruses. Small viruses present, highly divergent. They have not been associated with any disease whatsoever, but they're there. They probably evolved with humans and also animals in general over millions of years, so that part of our microbiome are there. We don't really know what they do, but they are. Then, of course, the champions of chronic infections are the herpes viruses. There's a whole range of different herpes viruses, which once they infect you, they stay with you for the rest of your life. Some other ones, polyomaviruses, causing warts or cervical cancer. Polyomavirus, also present in the blood. Circular double-stranded DNA viruses, which many of us have. Most cases, they don't cause any disease. Once you become immunosuppressed for whatever reason, they may come up and they can cause some disease, but most of the times they're there without even being noticed. And then, of course, some more familiar ones, HIV, hepatitis B, hepatitis C, all have the ability to chronically infect us. All right, so yes, of course, now with next generation sequencing being more and more accessible to everybody, this gives us a good tool to study the virome in much more detail. But before we can do that, we need to purify viral particles from, for example, stool samples or any biological samples. You need to do some purification. And the very simple reason is this: if you would just take a shotgun approach, you just take a biological sample and would just sequence every nucleic acid in there. Let's say there's a hypothetical situation. I have one cell of a bread wheat, one human cell, one E. coli cell, and then one virion particle of a number of different viruses. If we just look at their genome content, at the number of nucleotides present there, so for humans, for example, we normally have 3 billion, but of course they're double-stranded and we are diploid, so that gets to 12 billion. So that would be more or less the number of nucleotides present in each of these cells or viruses. So I would just extract all of this, do deep sequencing. Let's say, okay, I spend 100 million reads. This is probably what I would get for these different organisms. So the vast majority would be eaten by the bread wheat, the human cell, the E. coli cell, and quite a limited number of foul reads would be available through this approach. So we need to do some purification. We need to try to get rid of as much as possible of the bread wheat, human, E. coli, etc., all the cells, in order to get a decent view on the viruses present in our sample. So, well, you've seen this slide before. Actually, so with this in mind, we went to look in literature, and actually there are a number of different protocols in literature that use different ways of purifying or enriching biological samples for viruses, but none of these procedures actually look in detail what happened to a virus when I spin it, or when I centrifuge it, or when I put it through a filter, or whatever. So one of the things we did in our lab, we put together a virus mock community with these 10 viruses, and the idea was to encompass all the different characteristics of viruses there are. So I already mentioned some of the morphological characteristics, but there is also, we have, of course, RNA genomes, DNA genomes that can be linear, that can be circular, single-stranded, double-stranded, fragmented. So there's a whole range of different possibilities, which are all included in this mock community. And we put that mock community together, and again, the Mimi viruses we got very kindly from Pesaraú. So that we also mixed that with some bacteria, and then actually we tested a number of different procedures on this mock community to see what procedure would result in get rid of the most of the contaminants, being cells, human cells or host cells or bacteria, and keeping as much as possible of the viruses. So we did develop qPCR for all of these 10, and then tested all these different conditions with qPCR, how much do we lose, and I'm not going to bother you with the details of that, but doing that took us almost 2 years to do all of that. We came up with a protocol which we think is the best balance between trying to get rid of cells and bacteria and trying to keep your viruses. And this was published now at the end of 2015 in Scientific Reports. So shortly, what does the procedure do? Of course, so this is a schematic representation of a fecal sample with, in the different colors, different viruses, and in gray, different cellular organisms. So initially the procedure was developed on stool samples, but we've now tested a number of different samples, and it also works quite nice. So first you need to do, of course, is properly homogenize your sample. So we use a tissue homogenizer without beads, because we noticed if you use some beads, you will also destroy some of the viruses. Then we do things which are quite classical. You do a centrifugation step and a filtration step, but our centrifugation is quite mild, because also, again, we noticed that the procedures used in other papers, they use much more higher speed and longer temperature. You actually lose quite a bit of viruses. Also filtration, very often people use 0.2 or 0.45 micron filters, but actually we noticed that many viruses which are supposed to go through there actually did not go through, or we lost a lot. So that's why we also opted for a larger filter size, also for the giant viruses. There's still, we lose some, but at least part does get through for the metagenomics. So once that is done, theoretically you're left with your viral particles and with potential debris or free-floating nucleic acids. So to get rid of those, we do a nuclease treatment. And then, theoretically, you're left only with your viral particles, only with nucleic acids, which are protected by a capsid or by a viriome. Then you can do an extraction, and then we do a random amplification, converting all the RNA to DNA, library preparation, and deep sequence using Illumina technology. And then you get these millions of reads, and then trick is, sounds quite easy, put these genomes together, de novo assembly, and annotate them. I say that now very easy, but there are a lot of practical issues surrounding that, but I'm not gonna go into these. So this is a way that we now can use to study viromes in many different types of biological samples. So I say now the virome, but what does the virome actually encompass? What do I all mean with that? So of course the most obvious thing, yeah, virus infecting humans, the ones that are being investigated here on a very large scale. I was just shown around, it's quite impressive. So these viruses we all know about, these are common HIV or rotavirus or Ebola, whatever. Of course, bacteriophages, if you look at a stool sample, the majority of things you will find there, viruses you will find there are bacteriophages. Of course, infecting bacteria, archaea. We also can find viruses which actually infect other eukaryotic members living in your stool samples. Viruses that can infect fungi, parasites, protists, etc. And of course, in the stool sample, you can have viruses which are in transit, just from the food you consume, from the water you drink, from the air you breathe. There might be viruses in there which can end up in your sample as well. And then, of course, yeah, I mentioned before, some people consider endogenous retroviruses also part of the virome, but that's open for debate. So that's different types of virus you could encounter in these viral metagenomic data sets. So now, okay, we have these viruses, these different types of viruses, and how can these viruses play a role in our human health? As I mentioned, acute infections, we all know those, these are the most obvious. Integration into host genomes, not just as HIV does, and this can also have influence on the genome structure and might influence human genes present in the neighborhood of where the virus is inserted in the genome. Of course, bacteriophages, they might have a lot of interaction with the bacteria and indirectly influence our host. And then there are a number of ways which are probably less obvious, but also ways in which viruses could influence our human health. So these 3 points, for the sake of time, I'm not going to go into those, but I just want to give you a few examples of these other, probably less obvious ways that viruses can interact with our body, or we can cause differences in respect to human health. So the first one is something about interaction between viruses and other microbiota, which is often also termed as trans-kingdom interaction. So of course, you can imagine, yeah, of course, bacteria and bacteriophages, that's logic, they will influence each other. There also might be a number of other influences which are not that obvious. So for example, you would not immediately see a lot of interaction between bacteria and eukaryotic viruses. However, in many occasions now there's evidence becoming available that they are significantly influencing each other. So this is, for example, a schematic representation of intestinal tract where we have the villi, the mucus, and then of course all these microorganisms living here, and some of them trying to infect us. And for example, it's known for virus, for example for poliovirus, that when they interact with bacteria, they're much more stable. They're much more stable, for example, under heat conditions. So of course, in the gut, they have evolved for a long time with these bacteria, and they develop some interactions which benefits the virus in this case. Other examples is, for example, human norovirus. People have been trying to culture human norovirus for a long time. It's quite problematic. Now it's becoming clear that additional bacteria or certain bacterial components might help facilitate this infection of cells in cell culture. So again, bacteria are there helping the virus to achieve its goal. Other examples, MMTV, murine tumor virus, and they use LPS, also of course from bacteria, to change the immune tolerance of the host and therefore being able to cross those epithelial cells. Another one is that microbiota might also influence the interferon response, might influence immune response towards tolerance of these cells, so viruses can more easily infect. So a number of different ways that bacteria and eukaryotic viruses also can influence each other and can change the outcome of disease. Another example, so here it's kind of viruses that are helped by bacteria. Another example here is kind of the other way around, where bacteria are using viruses. So it's well known that in moments of increased influenza activity, that there's also an increase in bacterial infections, pneumococcal, staphylococcal pneumonia, etc. And what is kind of believed to happen there, how those two organisms, in this case eukaryotic virus and a bacteria, interact, is kind of summarized in this slide. So we have the respiratory epithelial here, the epithelial cells, we have the mucus layer, and when an influenza virus, in this case, infects these respiratory cells, one of the things it does, it has a neuraminidase which will cleave sialic acids, and thereby exposing particular proteins which can be used as receptors for other bacteria. In addition, when these viruses enter those respiratory cells, they are known to upregulate particular receptors for bacteria. They may downregulate antimicrobial peptides, so all things that happen that will give your bacteria more chance to actually also infect, also decreased activity of the cilia, to remove the mucus. So again, all these things together will allow bacteria an easier access to the respiratory epithelium and also for a systematic infection. So there's another way how eukaryotic virus and bacteria influence each other with respect to human disease. Another interesting finding, so these are two examples where viruses are kind of the bad guys, they cause disease. This is an example where virus actually might also have a beneficial effect to us. And this was a study done in mice. And as you might know, if you take mice and you deplete them from their microbiota, either by antibiotics treatment or by growing them into germ-free environments, they will survive. But if you look at their gastrointestinal tract, it will be very different from a healthy one. So you will see there that it's usually the gut, the gastrointestinal tract is shorter, the typical structure there, the villi, they're different. The immune cells normally surrounding the gastrointestinal tract are largely absent, so these mice will survive, but their gut tract is phenotypically really different. So what those researchers asked was, well, so it's known that actually the bacteria normally present have an important function in maturation, in obtaining a normal gut tract. So these people are wondering, well, can viruses also have such an effect? So what they did was actually they took some conventional mice with a normal microbiota. And when they treated them with DSS, it's dextran sodium sulfate, which is a chemical that normally causes injuries in the intestinal tract. But when the normal microbiota is present, these mice are fine. You will see after 20 days they all survive. However, when they now treated these mice right away from the beginning with antibiotics, so to make sure there's no normal microbiota, and then treat them with DSS, you will see that many of these mice, almost all of the mice will die after 10 days. So they will all be dead due to this DSS. So now they wondered, well, could the presence of a particular virus kind of restore the survival of these animals? So what they used was 3 different norovirus, murine norovirus, so norovirus that normally infect mice. And these 3 viruses were different with respect to the fact whether they cause an acute or a chronic infection. So this one up here, CR6, caused chronic infections in these mice, whereas the other ones had more acute diseases, so it would come and it would go again, whereas the chronic one would stay for a long time. And when they would then look at these mice, they would see that, for example, the mice treated with antibiotics and with the DSS, but chronically infected with one of these murine norovirus, that they actually did survive, almost all of them, after 20 days. For the acute ones, that was less obvious. So this first suggested that these viruses also might play an important role in the normal maturation of the gut. They also looked at some other markers, for example, the length of the gastrointestinal tract. I mentioned in these antibiotic-treated mice, this was shorter. So here they also showed that the infection with this chronic virus could actually restore this more normal phenotype with respect to length. They also looked at the number of immune markers, which I also said that were absent normally in these gnotobiotic mice, these mice without microbiome, and also these were to a certain extent restored. So actually providing the first evidence that viruses also may be responsible for a normal maturation of a normal gastrointestinal tract. So here viruses can actually also be beneficial. So another way that viruses, the virome, could influence our health is actually indirect through our host immune system. And well, I'm not an immunologist, but I want to just show you some very simple concepts which will be very useful to understand. A few examples I will give later. So of course, when a human, or any organism for that matter, is exposed to particular microorganisms, there is an immune reaction that will be generated. And here we divided in intracellular parasites, such as viruses or some certain bacteria or parasites, and extracellular pathogens, such as most parasites and bacteria. And when those infect a human body, they're being captured by antigen-presenting cells, and they're presented to naive CD4 plus T cells. And then they can either go to a T helper 1, they can mature to a T helper 1 immune response, or T helper 2. So T helper 1 is more referred to as a cellular immunity, whereas T helper 2 is more the humoral immunity, the antibody-based immunity. And one important, there's much more than that, but for sake of simplicity, I will stick to those two. One important thing is that they counteract each other. If one is stimulated, it will suppress the other one, and vice versa. So if you're exposed to something, you will go one way, inhibiting the other pathway. So as I mentioned, this T1 helper response, cellular immunity, will activate macrophages, will activate CD plus cells, cytotoxic T lymphocytes, natural killer cells will be formed, and they will cause inflammation in an attempt to control the infection. Whereas, as I mentioned before, for the T helper 2 response, you have more B cell activation and secretion of antibodies, so the humoral response, which then will fight invaders that way. All right, so with this in mind, I want to show you a few experiments. So the first experiment is with respect to helminths, that they might decrease the ability of an organism to clear a particular virus. So what they did in this experiment was they took some mice, and they know if we infect them with vaccinia, which is a pox virus, then normally the mice immune system is able to cope with that. You will have a relatively rapid clearance of the virus. So this is vaccinia. It's a virus intracellular, so will normally induce your T helper 1 immune response. However, if you, before the challenge with the vaccinia, so challenge these mice with a parasite, in this case Schistosoma manzoni, which is extracellular, so which will actually trigger the T helper 2 response, and you then infect them with vaccinia, then you see these mice much more problems, have a prolonged viral shedding, and they're not being cleared as easy. So here again you can see that different kingdoms of life, different types of organisms can actually influence the outcome of the other indirectly due to the human immune system. So it was further investigated, and it's believed that this observation is mainly due to a decrease of interleukin-2 and interferon gamma, and a decrease of cytotoxic T lymphocytes. So these are markers of the T helper 1 response, which are now suppressed by this parasite. A similar example, in this case a virus that might help us, is a herpes virus, which might protect against bacterial infection. So there's another experiment where they took mice, and normally when they challenge these mice with Yersinia pestis, which is an intracellular bacterium, these mice will die. These mice will not have the ability to fight off this bacterium, and they will die. However, if they first challenge these mice with a chronic murine cytomegalovirus, so intracellular T helper 1, virus intracellular, also T helper 1. So actually by giving the mouse this cytomegalovirus, you actually prime the T helper 1 response, and therefore the mouse is now much better able to fight off the infection with the intracellular bacterium, and therefore may survive. So again, this might be due to a prolonged production of interferon gamma and a systemic activation of macrophages, typical for the T helper 1 response, that might allow your mouse now to survive the infection with Yersinia pestis.
Just one question. How did you decide to choose this couple of bacteria and viruses? Is it true also for other viruses?
So you wonder if would the same be observed if I use other viruses? To the best of my knowledge, this has not been tested, but since you would assume that most other viruses also would trigger this T helper 1 response, you would also assume that, but to the best of my knowledge, this has not been duplicated for other viruses. Yeah, so if there's any question, feel free to interrupt me, no problem at all. All right. So another example of the interplay between our immune system and the virus, in this case in plasma virome, was an interesting study done in the UK, in the United States by UN de Vlaming. So what they were actually interested in, their laboratory, their hospital, they did a lot of transplants. so mainly heart and liver transplants. And of course, one of the things you need to do when you want to transplant an organ is to immunosuppress the people. Of course, you don't want to immunosuppress too much, because then people will be very vulnerable to all kinds of infectious agents. And you don't want the suppression to be too little, because then, of course, the organ will be rejected. So they were interested in these patients under immunosuppression, what would happen actually to the virome when these people are immunosuppressed. So this is one of the figures they generated. So what you can see here is day 0, this is the time of initiation of immunosuppression, and then a number of months after. And the black line is kind of the average of viral nucleic acids they find in the serum of these patients. So you see we have here heart and lung patients. Here there's quite some variability, but you see that there's quite a bit of increase of viral-like reads in the serum samples of all these patients, independent of what organ was transplanted. So the immunosuppression resulted in an increase in viral reads into those serum samples. And now they, of course, wanted to look, okay, what particular viruses were present there, were upregulated there. And there, this is what they found. So they did 2 cohorts. One cohort that also received antiviral drugs because they were CMV positive, and then a cohort which did not receive antiviral drugs, but they all received immunosuppressants. And what is immediately obviously here is this orange. So you see a number of different viruses identified in these serum samples. And in orange here are the Anelloviridae. You may remember a couple of slides ago that I mentioned these Anelloviridae, which are present in everybody's blood, not only in blood. If you would look in stool samples, in respiratory samples, you might also find them there. And they saw an enormous increase of these viruses present in those people which were immune suppressed. So again, I mentioned before, at this point, these anellovirus have not been associated with any disease whatsoever. So what this really means, that's not fully clear yet, but one of the things they're now considering is to use this viral load of anellovirus kind of as a biomarker for how well someone is immunosuppressed. To see, okay, can we use that to see do we need more or less immunosuppression? So again, this highlights that the virome, these viruses are chronically infect us, and our immune system are like in a constant arms race, or they keep each other in balance. But when, for example, like immunosuppression here, this balance is disturbed, then you can get a lot of these viruses that can increase in prevalence with potential additional effects to that. All right, and then the last way I want to present to you in which viruses can influence the outcome of health or disease is through regulation of host gene transcription. So the virome, of course, interacts with the human body, and of course particular genetic variations into the host might result in different phenotypes in one person to another. So you can imagine if you're being infected with one particular virus, normally what happens, your immune system detects the virus, initiates a number of transcriptional pathways in order to come up with an appropriate immune response. However, of course, if for example some mutations are present there in this cascade that results from a viral infection, of course, this might result in a different phenotype, in worse disease, if you cannot properly fight the virus. So that's quite obvious, a virus infecting a host. Another way this was shown is in a bit of a more of a complex example, and this was a study done on actually with the idea of better understanding Crohn's disease, which is an inflammatory bowel disease, chronic infection of the gastrointestinal tract, and they were studying this in a mouse model. And actually Crohn's disease is known to be partially genetic, so there are some susceptibility genes known, some genes and some mutations known that increase your chance of developing Crohn's disease in a human population. And they introduced this mutation into mice and then further studied what virus and the microbiome how that could influence Crohn's disease like phenotype in these mice. So what they found was actually, so what they used a wild type mouse, and then mice with this particular mutation in a gene ATG16L1. It's a homolog of a human gene for which we know if there are particular mutations, it will make you more prone to develop inflammatory bowel disease or Crohn's disease in this example. Again, they used a number of viruses. Again, in this case, chronic and acute virus. Again, this murine norovirus we've heard about before. And first, when they used these wild-type mice to infect with these chronic and with this acute virus, there was no particular phenotype in the intestinal tract. The mice looked normal. They had either an acute or a chronic infection, but nothing very specific to the phenotype of the intestine. Also, when they looked at the mutant virus infected with the acute one, the blue one here, the acute one, there was also nothing very striking happening. But when they infected the mutant one with the chronically infecting virus, they saw a Crohn's-like phenotype in the intestines of these mice, with some abnormal Paneth cells, cells responsible for the production of antimicrobial peptides, a unique gene expression profile of these infected cells, and also injury responses to the small and the large intestines of these viruses. And that only does so happen if you have a particular virus chronically in this type, in this example, and also with this particular mutation. In addition, what they did, they would treat these mice before they were challenged with the virus with antibiotics. So again, removing most of the gut microbiota, they would see again that the phenotype is normal again. So suggesting that not only the mutation, not only the virus, but also the microbiota itself plays a role in whether or not a phenotype of disease may occur. So here, so you need a chronic virus, you need a susceptibility gene, and you need the microbiota to get this CD-like, Crohn's disease-like phenotype in these mice. Whether or not similar things that happen in the human gut is still remains to be investigated, but at least it gives you an example of kind of...
Makes sense, because we've treated Crohn's disease with antibiotics since decades without finding a specific bugs associated with that, so it makes sense.
Yeah, so it's more complicated than that. It's probably multiple factors that play a role in this Crohn's disease. And actually, one of the things we're doing also in our lab is looking at the virome in these Crohn's disease patients to see, okay, is there anything, what is going on there? We know, for example, there's quite a bit of research done now for bacteria in inflammatory bowel disease, and there often they see when these people are sick, the bacterial diversity is much lower in these people. And actually for viruses, very limited data, and also things we confirmed that actually for virus we kind of see the opposite. In people that are sick, that have Crohn's disease, bacterial diversity is lower, but the viral diversity seems to be much bigger. Again, what exactly that means is still under investigation, but it's quite interesting. But again, in this mouse example here, it shows that different components might play a role in complex disease such as inflammatory bowel disease or Crohn's disease in this example. So a more recent hypothesis, which was kind of launched by Skip First in the United States, was the Virotype hypothesis with respect to viruses and effect of transcription in the host. So viruses, when a virus infects a host, when it infects a cell, it will alter the transcriptional stage of the host, not only the host, but also infect the other bystander cells by chemical molecules, by interleukins, etc., to notify other cells so they can also go into an antiviral state, for example, in case a virus is infecting the host cell. And many different types of host cells can sense the presence of viruses, and they can result with a response which is partially overlapping but is also distinct based on which virus or what receptor is triggered. So to give you an example of that, so this is a schematic representation of a cell, and cells have over the course of evolution developed a number of mechanisms to detect viruses, to detect pathogens. And probably the most well-studied class are TLRs, or toll-like receptors. And these are receptors on the cell that can detect various components of virus. For example, double-stranded RNA. Normally in a eukaryotic cell, you will never find double-stranded RNA. So when a detector detects double-stranded RNA, it will know, okay, there's something going on. There might be a virus infection or something going on. So what happens then, there's a whole cascade of enzymes which is being activated and which will result in differences or changes in the transcriptional stage with cytokines, chemokines being expressed, and this may result then in inflammation. These chemokines and cytokines may warn other cells in the surrounding in an attempt to limit the spread of the infection. So there are a number of other TLRs here which recognize different parts of viruses, either protein, particular folds, particular amino acids, or other types of nucleic acids that they can discriminate between the host and invader nucleic acids. And all of these have activate a cascade of enzymes that will result in this change in transcription. So depending on which virus, which part of the virus recognized, a slightly different response might go on in the nucleus of these cells. So that's on the cell surface. Similar receptors can also be present on the endosomes. And there's also a number of receptors that are intracellular, so in the cytoplasm, and which can also detect viral infections there, again, double-stranded RNA, or for example, APOBAC, which will detect foreign nucleic acids and introduce mutations in an attempt to stop the viral infection. So different viruses will elicit different reactions, different change in the transcriptional stage of infected cells or infected organism. And as I mentioned before, we're all chronically infected with a number of viruses, and you remember this slide. And this is just a slide with viruses that we know of. Probably there are many, many more than these, so probably each of us is probably infected with dozens and dozens of chronically infecting viruses, and they might be very different between you and this person sitting next to you. An additional thing that has intrigued people before is that autoimmune diseases very often seem to be preceded by an infection of some sort. People think, okay, this infection might have triggered some autoimmunity, but it has been very difficult to link a particular virus to a particular autoimmune disease. So that's kind of where the Virotype hypothesis comes in. So what do they mean by Virotype? So with Virotype they mean a group of viruses that share the same cell tropism and the capacity to activate the same sensors. That's kind of what they term Virotype. So it could be that different viruses, completely unrelated viruses, do have the same host cell as a target and do start the same response. So kind of Virotype means virus that elicit a very similar gene transcriptional change in the host. As I said, so these elicit a predictable and host translation response and may be responsible for certain disease. So it could be that some of these autoimmune diseases, we will not be able to find one particular virus, but we might find that particular groups of viruses starting the same cascade might induce some of these diseases. So to kind of summarize that and also kind of summarize what I've tried to tell you today. So if we have different people, all of us, we're genetically diverged, we have all different alleles, we might respond different to particular viruses. In addition to that, we all have our unique virome, which is can be very different from between two persons. So that will respond, this mix will respond in very different transcriptional stages in the cells we all have in our body. And of course, when you think about it this way, then if there might be particular triggers, external triggers that could, where you could be exposed to, this might lead in certain people to disease, and in other people this may not, based on this whole diversity of genes being expressed and the body reacting to that. So with that, I hope I have kind of given you a flavor of a number of different ways that viruses also can influence our health, in addition to the very classical ways we see viruses causing disease. So with that, I would like to thank you for your attention. I will try to answer any questions that you may have.
Something I'm really fascinated with is the role of the bacteria in facilitating the viral infection. So I'm sure you know this paper that have shown that poliovirus and enterovirus enter only if you get the bacteria into the gut, and,
yes.
and it doesn't work. I get two experience of this in one question. One is that when I was younger, treating Q fever for 7-8 years, doxycycline. When I stopped doxycycline, people were saying, well, during all this time, I've never get flu for 7 years. And then stopped doxy, and then I start to get flu one more time. And when we're looking now at the microbiota of the people with respiratory infection, it seems that the increase of the pathogen is coming very, very early. So we get plenty of Haemophilus, specifically Haemophilus influenza, Pneumococcus, and Staph aureus, very, very early. And the main difference in the population with the virus and symptoms and with the virus and no symptoms is the presence of this pathogen. So it is since the very beginning something. And something I'm really interested in what you show is that apparently you say that norovirus need to get the bacterium to multiply.
Yes, so it's people have tried for a long time to culture norovirus, and actually there was a study was done in it was actually by accident. So normally what you do, you take a fecal sample of a norovirus infected person, and people first try to filter that, just get the virus and put that in your cell culture. You don't want bacteria in your cell culture. It's gonna. But apparently at one time, I am not 100% sure it was the idea, but it might have been an accident. They put on a sample there where there was still a lot of the bacteria from the fecal sample present there. And actually, to their surprise, they did see some replication. They have tried, well, so...
And I'm going to tell you something. Are you growing virus?
No, we're mainly looking at the genome.
There is something that bothered me since years, and it's interesting, it's coming with this, is that the people who grow virus, you know, I started a long time ago as a rickettsiologist. Rickettsiologist is growing intracellular bacteria without antibiotics. We were the aristocracy of the intracellular bugs, because the virologists put plenty of antibiotics and they play dirty because they need the security. And now we have, I'm sorry that Bernard is not here, but now I want to go on culturing a virus without antibiotics. And we think, of course, we find out that some antibiotics are effective against virus. This is the first thing. And the second thing is probably, yes, of course, it's removing part of the partners of the growth. And I think that we probably have missed many of the virus just because we use systematically antibiotics.
Yes.
And do you think that norovirus could be prevented by antibiotics?
Probably, if you want to treat norovirus by antibiotics, I think you might do much more harm by using the antibiotics and destroying your bacteria rather than preventing the norovirus. So you mean preventing the norovirus by playing on the bacteria? You don't mean a direct effect on the virus?
No. Playing on the bacteria.
Yeah. So
this is what you say. I mean,
yeah.
At the end of this paper on poliovirus, this is what they say. I mean, you can prevent poliomyelitis if there is an outbreak by giving doxycycline.
So if you, well, it will depend. If the interaction between your norovirus and a bacterium is very broad, so meaning many bacteria could serve the same purpose, that would mean you really need to wipe out a lot of the different bacteria. If it's quite specifically, you could just knock out those couple ones. I think it could be useful if you have to wipe out a lot of them. That will also might have some additional effects you don't want, but could be an idea to play with.
Well, the other thing that we were discussing with Michel is that, well, maybe cytomegalovirus in human beings prevents plaque.
So that prevents plaque?
Plaque.
Plague, okay, yes.
Yes.
Yeah. So
the experimental model will show that. So if you get CMV, probably we don't have any more outbreak of plague just because there is an outbreak of CMV before.
could be, yeah. So this is just probably very much the tip of the iceberg. There's some experiments showing this. Probably there's much more of these interactions going on where we at this point are absolutely not aware of, but which might in certain cases be very important.
Makes sense. Questions? Moi je suis curieux, mais vous avez le droit d'être curieux aussi.
You said homogenization of stool samples with beads reduce the and restrict the viruses. What are the viruses?
So I showed the 10 VARs we selected, and of course we added that, we mixed that with bacteria, and then we tested different types of beads, different intensities of the shaking. And actually what in general we found that not all viruses, but some viruses are actually quite affected. So if we look at our qPCRs after homogenization with the beads, we lost quite a number of these viruses. So that's why we stopped using the beads for fecal samples. That's fine, you can just homogenize without beads. For other types of samples, of course, you have tissue samples. Yeah, you might need beads. Will be harder to do it without beads. So there you'll have to use the beads, but you should keep in mind that you might lose some viruses by that procedure.
What about the bacterial? You mean the qPCR or viral metagenomics?
No, so for the optimization of the procedure, we used qPCR. So we had these 9 viruses, and we had a number of bacteria, and then we had qPCRs for each of them to really see exactly what happens under particular treatments.
And doing the metagenomics of virus of human, for example, fecal samples, what are the main groups of so-called transitional viruses that you attribute to plant viruses, insect viruses, and so on? What is the proportion, and what is the main groups?
So the proportions, of course, when you look at the stool sample, the majority will be bacteriophages. Actually, the majority of what you will get will not be classified or misclassified. For example, that's a big problem with the bacteriophage annotation. There are a lot of bacteriophages which are present as prophages in bacterial genomes. These bacterial genomes are being sequenced, they're being annotated as bacterial. And when you blast or do any comparison of your phage to a database, it will come up, that's a bacterium, where it's really not. So in the stool sample, the majority is of course phages. And I think with our method, we do get also quite well rid of like food remnants. So we do sometimes see plant viruses, not that frequently. There are other papers that describe a lot of plant viruses. We don't see them that often. But we do sometimes see like partiti virus or virus which they very likely infect fungi or something. So that's probably either infecting the fungi living in our gut, or they might be from whatever food source. So the vast majority is still a bacteriophages. And then depending eukaryotic virus, if you have an acute infection, they may eat the majority of your reads. The majority might be that eukaryotic virus. If you have samples from an asymptomatic patient, eukaryotic reads are usually relatively low also. You might get some of the anelloviruses. And then yes, sporadically you might have some plant virus, some fungal virus, or some insect viruses. But the majority would be phages. But again, one of the problems is very often it's difficult to annotate them because of annotate as bacterial. And in addition, there's a lot of phages. Well, we're pretty sure they're phages, but they don't show any homology whatsoever to any known virus in database. So very often we have, for example, if like consecutive samples, and we find in each of these samples a counting of 40,000 nucleotides, high coverage. So that's not going to be bacterial, because you expect it to be bigger. You find it in consecutive samples. So we're pretty convinced they're phages, but they don't match to anything in database. That's an additional challenge. Even in the human gut, there is still a lot of viruses out there which are completely uncharacterized.
I have one question. So what kind of genes you target for qPCR
For the qPCR, the virus in the optimization?
Yeah.
Well,
there is no conserved genes, you say. So.
No. So we had a qPCR for each of the individual viruses. So if you just target one particular known virus that you put into your mock community, it doesn't even matter what you target. You just know what you're looking for, so you can just use pretty much any gene, design your qPCR. So we had a mock community of these 9 viruses, 4 bacteria, and we had an individual qPCR for each of them. and then we subjected them to different purification conditions, and afterwards we checked again with qPCR, and we could see a drop in viral load if particular treatments would result in the loss of particular viruses. So we targeted different regions for each virus.
Can us define some virus or some family virus that has some genetic virus that can give some benefic trait to a host, to a host, for example, like Rickettsia antibiotic that give some benefit to a host or
Yeah, so the question is, are there viruses that also might be beneficial to you? So I think, for example, that one example I gave you in the mice, where if you chronically infect the mice already with a particular herpes virus or cytomegalovirus, that it then allowed the mouse to better fight another pathogen. In this case, it was the intercellular bacterium. So I think, and also the other example was with the norovirus in the mouse without microbiome, that the chronic norovirus infection could to a certain extent restore some of the normal gut physiology in these mice. These are two examples, and probably there are many, many, many more which still need to be investigated. So it's probably just the tip of the iceberg. So it's very likely that many of these viruses, so most of the viruses you know are the ones that are pathogenic. These are the ones which are investigated. These are the ones where all the money goes to. But there are many, many more viruses which are probably there all the time, and they might have many beneficial effects where we are at this moment are not aware of. But they for sure will have beneficial effects as well of these viruses.
Okay, thank you again.